So in this section, we're going to be talking about T cell development. And like B cells, T cells also originate in the bone marrow, but they undergo most of their development in the thymus, hence the name T cell. So from the bone marrow, T cell precursors leave and enter the thymus. What we're seeing here is an electron micrograph of thymocytes, or the cells in the thymus, interacting with T cells, or thymocytes. Those are these little balls right here interacting with this tissue. After maturation in the thymus, mature T cells leave and then populate the secondary lymph tissues, like the lymph node, the spleen, and the Peyer's patches. Just like our lymphoid tissues, right, the lymph nodes in particular, the thymus has a very particular organization, and that helps to promote thymocyte, or maturing T cells, and their interaction with the epithelial cells of the thymus. So on the right, we have a cartoon schematic of this histological cross-section that we're seeing on the left. We have structural components, the capsule, and tuberculae, which encase a segment of the thymus. There's a cortical area just below the capsule, and we'll see that in this cortical area, thymocytes die a lot, right? So during this process, we waste a lot of our cells. A lot of these cells are not going to be fit to be functional immune cells. We also have Hassel's corpuscles, and these are important for releasing a lot of different cytokines that modulate dendritic cell activity. Their exact function isn't known, but within the thymus itself are embedded a lot of dendritic cells, and these dendritic cells derive a lot of their cytokine information from these corpuscles. Now, during the development of T cells, this mainly occurs when we're younger. As we get older, the thymus diminishes, and it undergoes a process called involution. So if we look at the number of functional thymus-making T cells, the majority of those are occurring before the age of 26 to 50 years old. And after that, the thymus, shown here in panel A, shrinks to become looking more like panel B. The organ itself begins to shrink, and our functional T cells aren't made as readily anymore. So most of this process is occurring while we're younger. So how is it that a T cell becomes a T cell? Well, it's a lot like the same way we discussed a B cell making a dedicated decision to becoming a B cell. Right? Each cell is exposed to a different kind of environment, which elicits changes in gene transcription, which imbue that cell with new markers, new proteins being expressed on the cell surface that we can measure. And that's how we define the different stages in lymphocyte development. Initially, we start off with stem cell surface marker, CD34, same way that we did with B cells. CD44, in this uncommitted progenitor, is also present before this T cell becomes a T cell. As it begins to mature, it expresses different surface markers like CD2, 5, as well as the IL-7 receptor, which we also saw in B cell development. But in this context, the IL-7 receptor is going to receive the same IL-7 message, but it's going to react differently, causing that cell to become a T cell. Additionally, we want to make note of CD4 and CD8. These are our T cell co-receptor molecules. And while the T cell is developing, it can sometimes be referred to as a double negative thymocyte. And what that refers to is it does not have CD4 or CD8. It's double negative for both of those markers. As that double negative thymocyte is maturing, it begins to arrange the T cell receptor genes in a very similar fashion to the way we discussed the B cell receptor genes rearranging in terms of their variable, joining, and diversity regions. Except in this case, we're just going to have V and J. So how do we know that these proteins are present on the cell, or if they're not? Well, we use FACS analysis in order to do this, or fluorescence-activated cell sorting. And we should revisit that video on the Blackboard website. In brief, this process allows us to measure the fluorescence or the brightness of different proteins by tagging them with a specific marker. So we can measure how much CD4 does a cell have versus how much CD8 does a cell have. If they have low levels of both, we call them a double negative thymocyte. If they have high levels of both CD4 and CD8, we call them a double positive thymocyte. In addition, we'll have those cells that only have CD4 and only CD8, and those represent our mature T cells. So how does this happen? What happens in the thymic epithelium that allows a T cell to begin this developmental process? So in the top of this figure, we have our thymocyte, or developing T cell. And in the bottom, we have our thymic epithelium, those cells to which those thymocytes are interacting. On the cell surface of these developing thymocytes is a protein called Notch1. And on the thymic epithelium is a protein called Notch ligand. When these two proteins come in close contact, a cleavage event occurs in which the extracellular portion of Notch1 is removed, which allows Notch1 to move into the nucleus where it can alter gene transcription. It displaces repressors that associate with the DNA, binds to a coactivator, and then initiates transcription. So before Notch1 was active, before this thymocyte interacted with this epithelial cell, there was no change in gene expression. After Notch1 was activated, it could bind to the DNA and turn on different sets of genes that are responsible for making a T cell a T cell. Again, imbuing it with a new function, an ability to interact with its environment in a different way. So before we go on, let's take a look back at the T cell receptor. Let's remember what parts of this T cell receptor there are and how it's similar to the light chain of the B cell receptor. If we see here that there's a transmembrane region to this receptor, these bind an alpha and beta chain to the cell membrane, so it's exposed to the surface, and they're modified with different carbohydrates. Now, the alpha and beta chain both have a variable region, just like the light chain of the B cell receptor. They also have a constant region in the same way. The variable region, if we recall, is going to interact with the antigen that is displayed on MHC. In addition, the alpha and beta subunits have to recognize MHC itself. In addition to the alpha beta subunits, we also have CD3, which makes up the T cell co-receptor along with CD4 or CD8. CD4 or CD8 is represented by this long extension here, this protein that specifically interacts with MHC class 1 or 2. If it's CD4, it interacts with MHC class 2. And if it's MHC1, it interacts with CD8. So if we were to start with an uncommitted progenitor cell, again we see our marker for pluripotent stem cell, CD34. A double negative committed T cell progenitor cell can no longer go back and become a B cell. It has changed the genes that it expresses, it has changed the proteins on its surface, and it can't go backwards. It's committed to becoming a T cell. And it expresses a protein called CD2. And if you recall, this is different from our B cell development, where they expressed CD10. These CD2 positive double negative thymocytes can then undergo rearrangement of their T cell receptor genes. And these are set up in a very similar way to the light chain during B cell development. So a T cell has a choice. They can become gamma delta T cells or alpha beta T cells. At this point, the genes for the beta, gamma, and delta regions are beginning to rearrange simultaneously, and it's a competition to see who can rearrange first. If the gamma delta region rearranges first, right, it's a different type of T cell receptor, beta rearrangement stops, a signal is sent to the cell, and that T cell becomes a committed gamma delta T cell. Now, we don't know much about these cells, but we do know that they don't require much MHC stimulation. Like they don't need MHC the way that normal T cells do, the way we've talked about them. And they have a very limited number of antigens that they can bind to. They're particularly important for fighting off infections such as Mycobacterium. If the beta chain rearranges first, that T cell expresses the beta chain of the T cell receptor, and this is an uncommitted double positive thymocyte, so it expresses both CD8 and CD4, and eventually it's going to have to make a choice. Which one is it going to express? And we'll get to that decision later. Upon successful rearrangement of the beta chain, again, there is a choice that the T cell has to make, and rearrangements of the alpha, gamma, and delta regions occur at this time. So if we look here on the right, we'll see why this choice is permanent. If the T cell rearranges a variable alpha with a joining alpha, it turns out that the delta chain locus resides in between those two sites on the DNA. So if this rearrangement is successful, we have the variable region of alpha joined to joining alpha, and the intervening DNA is excised and deleted. That means that we lose the delta locus, and that cell can no longer go back and become a gamma delta T cell. But at this point, the cell also has a second chance to become gamma delta T cell. If the delta locus rearranges first, it sends a signal to stop alpha rearrangement. And again, that cell becomes a committed gamma delta T cell. If the alpha region recombines first, again, it cuts out the delta locus and becomes a committed double positive thymocyte with an alpha beta T cell receptor. So from here, we're going to consider that most T cells are going to become alpha beta T cells. Gamma delta T cells are a rare subset, and we're going to be focusing more on these alpha beta rearrangements. So first, in the T cell, we have a beta chain rearrangement. And 2 attempts can be made at a productive rearrangement here, because just like the B cell receptor, we have multiple chromosomes where we can try to rearrange this information. Unlike the B cell receptor, we don't have various types of light chain, like variable kappa and variable lambda. So here, we have fewer opportunities for combinatorial diversity. Right? But our V beta is going to recombine with a DJ region that's already been successfully recombined. If that's non-productive, there will be successive rearrangements, just like in the B cell receptor development. Now, once we have productive rearrangement, we have a functional beta chain gene that can be transcribed into this mRNA, which is going to result in the synthesis of a T cell receptor beta chain. At this point, we have a checkpoint. We have an ability to see, did we successfully create a productive beta chain of our T cell receptor? And again, this happens in a similar way that we tested heavy chain rearrangement in the B cell receptor. In this case, we don't have a surrogate light chain, but we have something called PT alpha. And this acts as sort of like a surrogate alpha chain, in the same way that the pre-B cell receptor had the surrogate light chain. Now in this case, we have our surrogate alpha chain with our successfully recombined beta chain. And we have 2 copies of that. The checkpoint here is that these 2 pre-T cell receptors can form what's called a superdimer. This PT alpha interacts with this beta chain, and this beta chain interacts with this PT alpha, creating a dimer. When this dimer occurs, it recruits CD3, or the T cell co-receptor. If the beta chain is capable of inducing a successful signal, it will halt rearrangement and begin rearrangement of the alpha chain. So it's this superdimer, right, this cross-linking of the beta chain that's been produced that initiates a signal to indicate whether that beta chain functions or if it doesn't. If it functions, it then signals for alpha chain rearrangement. So upon successful completion of a beta chain, we initiate alpha chain rearrangement. And again, just like in the B cell receptor, this can have multiple attempts for productive rearrangement. So we'll start by attempting to join a variable alpha with a joining alpha region. If that's not successful, we can initiate a second round of rearrangement and a third by mixing and matching different regions from variable and joining segments. Once a successful rearrangement is completed, there is expression from the variable region down to the conserved region, and that RNA is spliced to create a protein that is a T cell receptor alpha chain. This can then interact with the T cell receptor beta chain. So if we take a look at this from a broader perspective, we have progenitor cells entering the thymus from the bone marrow, and they're encouraged to undergo proliferation. So they're interacting with cells within the thymus that are sending them a signal to proliferate. They then commit to the T cell lineage by expressing specific genes that allow them to just become T cells. So they can no longer become other kind of lymphocytes. Once they're committed to this lineage, the beta chains are rearranged to create a functional beta chain T cell receptor, and this is when we reach our checkpoint, the superdimer of our PT alpha with our new beta chain. Those that are successful in having produced a functional beta chain proliferate. And we call these immature double positive cells. So now they are expressing both CD8 and CD4. These are our T cell co-receptors, at which point they're going to rearrange their alpha genes for the alpha portion of the T cell receptor. And there again will be a checkpoint to see if we rearrange that alpha receptor correctly. If this does not happen, that T cell is going to die. So within the cortex of the thymus, we can see a lot of cell death. Many of these rearrangements are not productive. And there's an immense amount of waste of T cells as they're developing. It's why we need to have so many proliferation steps. If we start with very few cells, there's not going to be enough to survive this process, this selective process. After the checkpoint, we have a functional T cell receptor with an alpha and beta chain, both with CD8 and CD4. So we call these double positive thymocytes. We'll talk about this in a moment, but these then undergo positive and then negative selection to make sure that they are not self-reactive, and also to determine which type of MHC they recognize. if it's CD8 or CD4 interacting with MHC1 or MHC2 respectively. So what's going on in the cortex of that thymus? I said there was a lot of cell death there. How do we get survival of our successfully rearranged T cells or death of those that weren't very functional? So on the right here, you're looking at a histological cross-section of the thymus, the medulla, and the cortex. Each of these dots is a result of a staining. Right? So we've added a chemical to this slide, and anytime there are cells undergoing apoptosis, they show up as these brown spots. So these are foci of cell death, which means that a lot of cell death is occurring in the cortex of that thymus. So what does that mean in terms of selection? If the TCR that we've created, we've recombined it, and it's functional, fails to interact with an MHC molecule on epithelial cells in the thymus, that thymocyte dies. And that's what these represent, these spots here. If that alpha-beta subunits of that T cell receptor are capable of interacting with MHC and its antigen through moderate or strong binding, that T cell is given a signal to live. Right? And that makes good sense. We want a T cell that has a functional alpha-beta receptor. We know that the protein works. And now this step, we're confirming that that T cell receptor can recognize MHC and an antigen. So at this point, we have double positive thymocytes, and we know that they can recognize MHC and they can recognize an antigen. So how do we become mature T cells? Either CD8 positive or CD4 positive. Well, in the thymic epithelium, there are cells that express both MHC class I and MHC class II. And each of those is going to be displaying different antigen. If our double positive thymocyte interacts with MHC class II, MHC class I, CD8 is going to bind to MHC class I and produce an intracellular signal. Right? So CD8 binds to MHC1, and that stops production of CD4. And now this is a single positive thymocyte. It has one T cell co-receptor and one T cell receptor. On the other hand, if the T cell interacts with MHC class II, CD4 is going to bind to that molecule. That will send a different intracellular signal inside the cell that will stop production of CD8, leaving us with a CD4 single positive thymocyte. Now this cell can go off and perform the effector functions of CD4 positive cells after it's gone through negative selection. So how does negative selection occur in this case? So right at this point, we have a single positive thymocyte, and we're going to test how strongly it binds. If we have moderate binding, that cell lives. It's given a signal to survive. If there is very tight binding, that cell is given a signal to die. Now, if you think about that, that might be a little bit confusing. Don't we want our T cells to bind tightly to antigen? Don't we want them to recognize MHC well? But let's think about it. What happens during tight binding? What are we actually talking about? Every time this surface protein, this T cell receptor, interacts with its ligand, MHC antigen complex, it delivers a signal to the cell, right? An activating signal. If that signal is very strong, that could result in T cells that inappropriately activate themselves. They might not need the signals like cytokines from their antigen-presenting cell. And that could result in aberrant activation of T cells, which could lead to an autoimmune disorder. We certainly don't want that. So we want our T cell receptor to interact with its ligand, but not so tightly that it causes activation inappropriately. So if we take a look at this from beginning to end, We start off with a proliferation followed by a differentiation into double positive thymocytes. Right? They're going to be in the subcapsular region. During positive selection, double positive CD3 positive thymocytes are in the cortex, interacting with cells in the thymic epithelium, as well as dendritic cells. Those that proliferate undergo negative selection, and our double positive CD3 positive thymocytes are found throughout the cortex, especially in the junction between the medulla and the cortex. So after vetting of these, we become either CD8 or CD4 positive, but not double positive. We know that they do not recognize self-antigen or are inappropriately activated by just binding to MHC. Now these vetted T cells with either just CD4 or just CD8, but not both, leave the thymus in the blood and re-enter circulation of the lymph through entry through the thoracic duct. They then go out and populate the secondary lymph tissues like the lymph node, Peyer's patches, and the spleen.